touching on the polysulfide molecule in cardiomyocytes, I would like to briefly talk the background of my study. Okay, so I first considered the relationship between my study and acupuncture. So the common mechanism is focusing on the sensing the proteins in the cardiomyocytes. So because acupuncture is reported to have the many beneficial effects on my body, so anti-pain and anti-inflammation and facilitating the muscle flexibility and neurohumoral homeostasis. So the common mechanism working on the cells and tissues, some TRP channels, transient receptor potential channels, may be the very good target of the acupuncture and our drug discovery and the treatment of the heart failure. So we focus on the mechanoactivated TRP channels in heart. So this TRP channel is very famous for the Nobel laureate by David Julius, and also first identified by the Craig Montel as a phototransduction mutant in Drosophila. So okay, so we work on the mechanism of the redox coupling in heart. So look at the diastolic storage induces the loss production in heart. This is very important to enhance the cardiac contractility, but the molecular mechanism was unknown. In 2011, Redler's group in the US has first published the plasma membrane NADPH oxidase NOx2 is work as a mechanoactivated proteins to induce the loss production, and so this loss oxidize the ryanogene receptor 2 to facilitate the calcium-induced calcium release. However, the NOx proteins, this NOx protein can be only activated by the protein kinase C and calcium. So I think that some so calcium permeable channels may be involved in the mechanical storage-induced NADPH oxidase activation. So I focus on the mechanoactivated channel TRPC3 and TRPC6. Okay, so I first saw that these two channels, TRPC3 and TRPC6 channels, are diacylglycerol activated channels, and they cooperatively work on as a calcium permeable channels to activate the calcium-dependent signaling pathways to induce the cardiomyocyte hypertrophy and heart failure in the heart. However, so first we obtained either the TRPC3 and TRPC6 knockout mice, and evaluate the cardiac functions after the pressure overload models. So the result was contradictory. So quite different. When the knockout of TRPC3 can preserve the cardiac functions after pressure overload. However, TRPC6 knockout failed to improve the heart failure after pressure overload. So these two channels are functional, very similar functionally, and the amino acid sequence is very similar. However, the output of the response is quite different. So we next think about the mechanism, what is different between the TRPC3 and TRPC6. So we noticed that TRPC6 channels specifically permeate the metal ions such as zinc and ferrous. And previous papers suggest the deletion of the TRPC6 channels perturbs iron and zinc homeostasis and pregnancy outcome in mice. So I haven't believed this report, but the result was very similar to our work. That's why we hypothesized that this TRPC6 mediated zinc influx is important to enhance the cardiac contractility in the heart. Okay, so zinc is a very trace amount. It's very difficult to visualize in cells and tissues. So yeah, actually the 10% of the zinc is present in membrane, but so totally the free zinc levels are about the picomolar to nanomolar level, very very low compared to the calcium and sodium. So it's very difficult to identify. So there are the only limited technique to visualize the zinc levels. So, but zinc is actually very important to keep our health. So, so I so really enjoyed eating the Korean foods such as the oyster, the beef, and the liver. So during 2 days, so these foods contains a very high amount of the zinc, and this is very important to maintain our homeostasis. So okay, so the deficiency of the zinc may cause the hair loss, taste disorder, and the immune dysfunction and diarrhea. And so the heart is also the case. So zinc deficiency exacerbates the stress-induced myocardial injury, and plasma zinc concentration is positively associated with heart failure severity. And although so in Japan, also the case. So although the zinc supplement is not clinically approved for the treatment of the patients with heart failure, so some cardiologists, so especially in the Kyushu University Hospital, experimentally so recommended heart failure patients in the hospital to take the zinc supplements. So this is the background. So yeah, we first analyzed using the trypsin-6 knockout mice, which can permit the zinc through the plasma membrane. So yeah, we treated the zinc deficient diet to the wild type mice trypsin-6 knockout diet. So before the treatment of the zinc deficient diet, so the cardiac function was the same between the wild type and trypsin-6 knockout. However, after 4 weeks, the treatment with the zinc deficient diet, only trypsin-6 knockout mice shows a severe reduction of the contractile functions compared to the wild type. This may be the high motivation to me to promote our work. So the next, what is important for the, we next analyze this zinc influx may be required for the baroreflex or dependent cardiac contractility. Baroreflex response is very important to maintain our cardiovascular functions. However, when we use the hydrolyzing, this is the experimental reagent to transiently reduce the blood pressures. So when we treat the mice with this hydrolyzing, so the blood pressure is transiently decreased. Then the baroreflex dependent cardiac, increase in the cardiac contractility was observed in a time-dependent manner in the wild type. However, in the trypsin-6 knockout mice shows a nice reduction of this contractile responses. In contrast, the heart rate was not changed between the wild, was not affected between the wild type and the trypsin-6 knockout mice. So, okay. And then we further analyze the molecular mechanism. Maybe the sympathetic nerve dependent noradrenaline release is involved in this passive reflex dependent increase in the cardiac contractility and heart rate. So we next analyze the involvement of the beta adrenergic receptor, expressing in the heart cardiomyocytes to analyze. The result was very similar to the previous slide. That in the trypsin-6 knockout mice shows a nice reduction of the baroreflex and the isoproteinol treated increase in the cardiac contractility as shown in the wild type and the trypsin-3 knockout heart in the blue line. In contrast, increase in the heart rate was not affected among all groups. These results suggest that trypsin-6 knockout mouse shows that zinc dependent, maybe a zinc dependent reduction of the cardiac contractility. Okay, so we further analyze the molecular mechanism. Why the zinc enhances cardiac contractility in hearts? So we luckily identified the nice structure of the zinc, the localized between the GPCR and heterotrimeric G proteins, especially the beta adrenergic receptors and G alpha S, GS trimeric GS proteins. So this coupling, the requires the zinc to the enhancement of the GPCR and G protein coupling. But before publishing this result, we failed by the European group. First published this paper in the Nature Chemistry, and this is completely the same result. We are very shocked, but it's okay. Yeah, and yeah. Finally, to make the story very short, before touching on the sulfur biology, we found that this TRPC6 mediated zinc influx may facilitate the beta adrenergic receptor and GS protein coupling. And this enhancement of the G protein and GS protein coupling facilitates noradrenergic receptor mediated cyclic MP and response and increase in the cardiac contractility. This was already published in the Nature Communications in 2022. We further analyzed whether the activation of this TRPC6 mediated zinc influx is beneficial for the treatment of the chronic heart failure. We had a very positive result to improve the chronic heart failure after pressure overload or myocardial infarctions. We next analyzed the molecular mechanism. Very surprisingly, this compound, TRPC6 activating compound, PPZ2, can very strongly inhibits the oxidative stress during the process of the chronic heart failure. We focus on the intracellular redox homeostasis, carefully looking on the sulfurs. This is a probe for the selectively visualizing the polysulfur molecules, sulfane sulfur molecules. When we use zinc chelators, this sulfane sulfur molecules levels are highly decreased. This can be we found that this polysulfide, we call this supersulfide. This formation is mediated by the zinc influx in the chronically to improve the chronic heart failure. What is the supersulfide or polysulfide? Actually, in the textbook, we know that the cysteine or glutathione or cysteine or oxidized glutathione, these molecules contain the sulfur molecules, atoms, and this is very nucleophilic properties. However, when this sulfur molecules can be connected to each other to make the so-called catenated forms, cysteine persulfide, trisulfide, or tetrasulfide, and also this molecules has a very strong moiety to have both the electrophilically and nucleophilically. This is a very unique profile compared to this cysteine or glutathiones. Okay, also these proteins supersulfide is not a formal terminology, but how to say it's just a slang terminology. But this supersulfides contains all polysulfide molecules in the heart. And this polysulfide, the so-called supersulfides, are highly expressed in mitochondria. And this supersulfides are very important to maintain the energy metabolism and stress response and adaptation in the cells, and membrane and potential formation, and also the quality control of the proteins and also the organelles. And also in the heart, considering the clinical application, maturation of the cardiomyocytes, human iPS-derived cardiomyocytes is now a very current topic. Actually, iPS-derived cardiomyocytes is immature and it's very difficult to maturation. So yeah, during the process of the maturation, the metabolic, the reprogramming of the mitochondria is very important, as shown in this review papers. So how to maturate the mitochondrial activity? So this is well correlated with the zinc permeability and the sulfide formation. So can you see the moving of the cardiomyocytes here? Yeah. So we facilitate the electrical pacing of the cardiomyocyte. This you can observe that this contraction and relaxation cycles as shown here. Yeah, we can see the contraction velocity and reaction velocities. And after the electrical pacing, more than 1 week, the cardiomyocyte structure is largely changed. So this is the alpha-actinin expression levels are highly increased, and the connexin 43 is also increased. And also we measured the intracellular zinc levels using the various artificial experiments using the fluorine-3. So fluorine-3 is a zinc selective chemical probes, but it is very difficult to observe the zinc dynamics directly. So we first release the all protein-bound zinc using the DTDP. And then, the zinc chelator was treated, zinc chelator T-pen was treated to goes down the intracellular free zinc levels. So the estimated total zinc levels were increased as the pacing of the cardiomyocytes. So we next evaluate the intracellular polysulfide sulfur and sulfur levels. So using the the sulfur selective probes. One is hydrogen sulfide anion, so H2S or HS minus fluorescent probes, SF7. And the second one is a QS10, which is selective for the sulfur and sulfurs, SS forms. So using these two probes, we found that compared to the cell lines such as the HeLa cells, the treatment, exogenous treatment with the Na2S, H2S donor, preferentially converted from the H2S to the sulfur and sulfur monomer, SS forms in cardiomyocytes, but not in the cells, cell lines. Okay. Okay. And also, we newly found that this sulfur and sulfur, the supersulfide formation, can be enhanced after electrical pacing for the 30 millivolt or 20 millivolt, as shown here. So there, yeah. These results clearly suggest that the zinc influx through the plasma membrane may facilitate the conversion from the hydrogen sulfide to the sulfur and sulfur molecules in cardiomyocytes to enhance the maturation of the cardiomyocytes. Okay, so yeah, considering this, based on this observation, we speculate that sulfur catenation in cardiomyocytes is very important to memorize the mechanical storage or the redox, changes in the redox homeostasis in cardiomyocytes. Because the loss or nitric oxide is very highly reactive and very easy to eliminate. So this can't be explained by it can't explain as working memory in cardiomyocytes. In the cardiomyocytes, pacing, repetitive pacing is very important to the continuous maturation of the cardiomyocytes. That's why the conversion of the loss signaling to the sulfur catenation may work as a working memory of the heart. Okay, so this is an in vitro experiment. So we then analyzed the in vivo analysis. So this was the maturation of the heart, the heart in the mouse. So the mouse has also the property of the metabolic reprogramming during the development. So we've successfully visualized the intracellular sulfur and sulfur levels using the another probe, STP1. So the immature heart shows a very the low signals, very weak signals at the 7 days after birth. However, in the matured heart, 8 weeks after birth, the sulfur and sulfur levels are highly increased compared to the immature heart. So we hypothesized, and so now published several papers to demonstrate the plausibility of our hypothesis that so in a normal heart, so the sulfur molecules are highly catenated by the redox-dependent mechanism. In contrast, so when so exposed to the ischemic stress or environmental so electrophile stress, such as metals and so aldehydes, so these so sulfur molecules can capture this so molecule, these electrophiles, and so highly so the forms so catabolize the sulfur molecules are accumulated in the heart. So this the accumulating heart is very vulnerable to the physical, so the mechanical load. That's why so to the increase the risk of the heart failure. To demonstrate this possibility, so we further focus on the several molecules which can induce the supersulfide formation in cardiomyocytes. There are 5 molecules such as cysteine, cystathionine beta synthase and cystathionine gamma lyase. Using the cysteine to generate cysteine persulfide in skeletal muscles. However, these 2 enzymes are hardly expressed in the heart. So we further analyzed and found that the mitochondria localize the cysteine tRNA synthetase. Cas2 is a key enzyme to generate cysteine persulfide using the 2 cysteine molecules. And also, so the sulfide quinone oxide reductase is another enzyme to convert the hydrogen sulfide to the supersulfide molecules in the mitochondria. So yeah, we using this the fluorescent imaging techniques further analyze in mouse models. So after the 15 minutes ischemia followed by the 24-hour reperfusion, so the supersulfide levels in the heart is so dramatically reduced compared to the sham groups. So we directly add back the inorganic NHS4, the supersulfide, to the cardiomyocytes, so that this supersulfide levels are highly recovered and the cardiac function is also recovered. In contrast, one cysteine persulfide generating enzyme, Cas2 hetero knockdown on the knockout the mice shows a severe reduction of the supersulfide after ischemia reperfusion, and the contractile function also goes down. This is well correlated with the supersulfide metabolism. So finally, so we aimed for some clinical application of this the supersulfide metabolism for the treatment of the heart failure. So yeah, we previously published that mitochondrial hyperfission is involved during the process of the chronic heart failure after myocardial infarction. So this process, mitochondrial fission, facilitates the early senescence of the cardiomyocytes, as shown here, visualized by the senescence associated beta-galactosidase staining. Okay, just speak. And so we look for some endogenous supersulfide molecules which can improve the cardiac functions after myocardial infarction. So yeah, exercise is known as a very beneficial for and improves the prognosis of the heart failure. So we treat the mouse with voluntary exercise for the 2 weeks. So we saw analyze the intracellular blood supersulfide molecules, and so luckily found that oxidized glutathione is highly increased after exposure to the voluntary exercise. So yeah, actually you know that reduced glutathione supplement is very beneficial also maintain our skin health. However, so this reduced glutathione never have the beneficial effect on the heart failure. However, so when we use the GSSG treated to the mouse after the myocardial infarction, this cardiac function is gradually decreased, but the GSSG treated mouse shows the recovery of the cardiac partially recovered cardiac function, as shown here. So we further analyzed the molecular mechanism and found that several the proteins, such as mitochondrial fission accelerating molecules, DRP1, has a critical redox active cysteines. These cysteines has a polysulfide modes basically. So polysulfide polysulfidated cysteines preferentially bound with this oxidized glutathione to make the glutathione stable glutathione of the proteins. This is the underlying mechanism how the GSSG improves chronic heart failure after myocardial infarction. Okay, finally, these papers are supported by so many collaborators. So especially I greatly thank the Professor Yoshito Kumagai, but not here. But he told me the the background of the redox biology here. That's why we successfully published this work. And also, I also thank Professor Sang-Eun Kim during the 5 years to supporting our work. Thank you very much for your attention.